Ready to start? Good. So I'm here to give you your ecology lesson. So my research has been in the development of quasi-closed ecological systems for deep space environments. I began this research 3 years ago now in regards to developing these systems for space habitation and for deep space environments. And so this may be your first, or maybe you've heard about closed ecological systems a lot, attending different conferences or just being curious about the history of space habitation. But I can say that many children have actually experienced and built closed ecological systems before. And you may have helped a child do this. You just take a 2-liter soda bottle, you put the dirt, you get the plant, you get the water, the air goes in there naturally, you seal it up and see what happens. So the idea of closed ecological systems isn't a new idea. In fact, the mention of closed ecological systems goes back to 1895 when, I have my notes over here, when Konstantin Tsiolkovsky published his book, Dreams of the Earth and Sky, in which he depicts and details a CES on a space station. And I'm in the process of actually reading this book, and it's a fantastic read. I would highly recommend it. And so from that point on, we have this vast history of closed ecological system research for space habitation. And I have the timeline here. So you can see that there's a lot that's been done. And so some of these closed ecological systems you may be aware of is the BIOS experiments done in Soviet Union Russia beginning in 1964. Biosphere 2 in Arizona with their first test module done in 1987. And then you have the Melissa project with the ESA, which commenced research and development in 2005. And then you have Japan's Closed Ecology experimental facilities, which started research in 1994. And then you have what my company, Deep Space Ecology, is working on, which is our 3-zone model. And if you weren't there at my talk yesterday, the 3-zone model, with what its name implies, has 3 different zones. The first zone is the habitation zone, where humans live, work, and go about their daily lives. And then you have the agricultural zone where humans can grow their food. And then beyond that, you have a wilderness or an ecological buffer zone, which allows for stability throughout the system. And if you want to learn more about the 3-zone model and about the research that I did to develop this system, you can check out our Deep Space Ecology YouTube channel, where we have past talks and YouTube videos depicting this. And so I can talk a lot about these ecosystems and how great they are, but you have to consider the problems that come with trying to close off an ecosystem. You go out here on Earth, you have all these ecosystems that are open to the air, they're open to each other, and they have had all this time to develop and become stable and resilient. So when you try to take that and close it off from everything outside of it, you're going to experience problems. One such problem is atmospheric fluctuations and the introduction of volatile organic compounds. Some of the engineers in this room might be like, well, you can fix this problem by doing a bleed and feed system, where you can use the outside environment to help with the atmospheric fluctuations and to help stabilize that. So you could create a technological scrubbing system to do that. But then there are problems that technology can't necessarily fix, such as out of control biological activity of microbial populations. This is one thing that happened in Biosphere 2, which led to a lot of the ecological problems. It also happened in Japan's facilities. They actually had to close down after 2 weeks after opening because they didn't take into account the rapid growth of the microbial populations. And then other problems are the lack of essential elements present in active circulation. And a picture that comes to my mind when I say this is the picture released by NASA from The Martian. You have the scene with the potatoes growing out of the Martian regolith, and you have the question, can plants grow on Mars? And then it has the list of essential elements where it's like, yeah, we have all of these available on the Martian surface. However, what they don't tell you is that even though, yes, those are present on Mars, they're not in bioavailable forms that plants would be able to actually take up. And that's important to consider, because if you go up there with this notion of, oh yeah, we have everything we need for plant growth, and you start planting different plant species, they're not going to do well because they're not going to have these essential nutrients that they need to grow because they're not in a bioavailable form for them to take up. So you have this lack of essential elements in active circulation. And then, of course, the overall degradation of the soil fertility with this. And then, of course, all these other problems, such as a lack of balance between supportive and competitive redundancy, unknown and known biological vectors being in the system, lack of biodiversity, increasing risk of disease spread and plant death, particularly when you don't have a high biodiversity there. And then, of course, the interactions between biology and technology. With Biosphere 2, they had the interaction between the atmosphere and the curing cement, which took the oxygen out of the atmosphere, and they hadn't accounted for that, and that led to one of the reasons why Biosphere 2 had to be opened. And then, of course, lack of water, lack of efficient water recycling, nutrient buildup in water systems that can lead to eutrophication and non-use of that water, etc. And so the important thing to consider with these problems is that it is hard to predict what disruptions will occur and the pathways that the reaction will take, such as, again, with Biosphere 2 and with Japan's system, the increased metabolism of the microbes with that biological material present led to a rapid increase in CO2, and the system didn't have the stability or the resilience to correct itself before everything just went downhill. And so when I was looking through this history of these systems, I saw a lack of applying ecological succession, which is the process that ecosystems go through to actually go from bare rock to a climax community like what we see outside. And being an ecologist and an environmental scientist, I'm like, well, that would make sense then why you would have these problems in these systems. These systems haven't had the time and the ability to create these biological cycles, nutrient cycles, and feedbacks that would allow it to be able to pretty much bring itself back from disaster if a disruption were to occur. And so what I've been doing with deep space ecology and developing these systems is applying this concept of ecological succession, how we can go from this barren, sterilized environment and go through a system of doing certain plant species at a time, certain microbial species at a time, to help develop these biological feedbacks and cycles that would allow it to have this stability and decrease the problems that we would be seeing. Of course, you can't get rid of the problems. There's always going to be problems, and there's going to be problems you didn't even think about. But this is to help make these systems possibly more stable and to increase the viability of creating an Earth-independent system. And so we head to Mars, and in the future, near or distant, we start to have these systems pop up. And if we look into these systems, they could look as good as this, like walking through a garden on Earth or being in an agricultural system. And with these, you have the biological side of things, but as you can see, you also have the technological side you have to consider. And so there's this dome structure that you're seeing. And so this dome structure is pretty much, it seems impossible, especially for a first go. And let me explain to you how we could go about building this structure so that we could house an ecological system in it. So this is the Mars EPIC X1 model that my company Deep Space Ecology designed. And my company's assessment is that the size of a system following this model required to support a team of 8 humans is 22,500 square meters, which is pretty big. And setting up a CES this size for a first mission to Mars seems unrealistic. However, considering the following approach of our Mars Epic X2 design, let's go about doing that. The mission's habitation zone is comprised of modules with fully engineered ecosystems that the humans have lived in since leaving cislunar space, and comprised of modules with fully engineered ecosystem, all of that, since leaving cislunar space, and expandable central habitation areas to connect everything together. Keeping up with my notes, I'm getting ahead of myself. And provide a larger living space. While initially living off of growing food and hydroponic systems in the habitation modules, the mission harvests in situ resources of water and carbon dioxide from the Martian environment, and builds the structures for the ecological buffer zone one section at a time, and harvesting carbon dioxide from the Martian environment, building these structures for the ecological buffer. And as the construction materials become available, you can start to make the system larger. As each section is built, the space would be pressurized with carbon dioxide and the regolith prepared using sterilized human waste, microbiological inoculants, water, and tilling prior to planting 1,000 plus species of seeds in a scattered fashion. Finally, as the structure is expanded, the sections of wild growth in the orange would be tilled under to provide an organic litter treatment for planting the food crops. So this could be a way to go about building a larger system over time instead of trying to do it all at once, and applying ecological principles to go about doing that. And so my experiment, I actually ran an experiment at Duke to test different pre-treatments that I could use for the growth and development of plants under Mars regolith simulant conditions. So the main point of this experiment was to see if plants could even grow from a stimulant that was completely sterilized. And this resulted in a double honors thesis, with the title of that thesis being The Development of Ecological and Environmental Mechanisms in Sterilized Mars Regolith Simulant to Drive Primary Succession in the Establishment of a Closed Ecological System on Mars. You just have to have that long title, you know? The initial title for it at the very beginning was The Development of Pretreatments for the Growth and Survival of Crops Under Mars Regolith Conditions. Yep. This became, you have to make it fancy for the paper, you know. And this is a picture from that experiment, and I'm going to go into some details about that experiment and the results that came from that experiment. And so the objectives of this experiment were to understand the mechanisms and variables that need to be present in the ecological and biological succession of a Mars regolith-based system for the eventual development of a CES on Mars, and to test the viability of growing plants and introducing microbial communities under Mars regolith conditions. And so the location and environmental conditions, this was done at Duke University's research greenhouses in Durham, North Carolina. The isolation zone that I was in was kept at a constant 21 degrees C with dehumidifiers to help control the water in the air. The hours of daylight were controlled using man-made light systems when we went into the fall season, and then this experiment started June 1st, 2016 and ended December 20th, 2016. And so the different regolith pretreatments that I started off with were an initial water contact condition, organic litter presence, microbial inoculation with biosolid application, and then different plant combinations. And so you can see here, that's what the Mars regolith simulant looked at like at the beginning. And I got that Mars regolith simulant donated to me by Sayar Industries out in Napa, California. They gave me 2,000 pounds of it for free. That was, and I had to mix it all myself because they gave me basalt sand, 1/4-inch basalt sand, and then baghouse dust. And the simulant requires 75% sand, 25% dust. And you touch that dust, it's poof. It was an interesting experience. And so the different plants that I was looking at were golden peas, Sonora wheat, broccoli, rapeseed, and French sorrel. And my main crops were the golden peas, Sonora wheat, and broccoli. And then the plants that I was using for a litter component were the rapeseed and French sorrel. And all of these plants were chosen based off of criteria that my advisors and I had come up with for whether these plants would be able to survive in such an environment, what are the multifunctional aspects of them that would contribute to the ecosystem as well as human use. For example, the French sorrel, you can not only eat it as a food crop, but is also a medicinal plant that you can use to help with upper respiratory infections. And then the microorganisms I was looking at were mycorrhizal fungi and rhizobium bacteria, and then of course the biosolids. And so this was the experimental design, where there were 120 pots, 1 gallon pots. 12 of them were in earth condition, making it 132. And then half the experiment was oven baked regolith, so there was no water content, no living organic organisms in there. And then the other half was steam sterilized to not only get rid of all the microorganisms, but to add a water component as well. Whether you have that water component, what does that do for you? And so that's how it started off. And then the results of this experiment. So I used ANOVA statistical analysis to look at how these different pre-treatments interacted with dried biomass production. And so in pots growing wheat, I saw that the microbial inoculation and biosolids application had a very good significant effect. And with this picture right here with the box plots, you can see on the top one that's between pots with microbes and biosolids on your left, and then on the right is pots without microbes and biosolids. And you can see that there's a wide difference in just the different data points that created this wide spread, and then a higher median, of course. And this is actually expected. When you are creating, when ecosystems usually form, you're going to have a lot of fluctuation in biomass production when you first start off doing that, because you're in competition, you're in supportive relationships, things are not stable at the beginning. And then another thing I was looking at was the soil respiration, which was where I was measuring percent CO2 that was being produced by the microorganisms in the Mars regolith simulant. And again, this is with pots containing wheat. And so the pots given the microbial and biosolid application pre-treatment is in blue, and pots without that pre-treatment are in orange. And you can see that the pots that had the microbes and the biosolids had a higher overall soil respiration and had it increasing over time. And this is important to notice because one, the microbes survived. I was worried about that. And two, they're actually increasing in population, which is what you want to see. You don't want to see that negative trend where it just stands still. This allows, this is important to determine whether the ecosystem will work. And then I was looking at the carbon-nitrogen ratios in the Mars regolith simulant, with the black bar representing the Mars regolith simulant control group with completely sterilized regolith, nothing added to it. And then these are different samples that I took from the different pots. And so the ones in blue are the pots given the microbial and biosolid application, and then the ones in orange didn't have this application. And you see that the carbon to nitrogen ratios are much smaller in the ones with the pre-treatment, and it's because these microorganisms are making the nitrogen available to the plants, and the biosolids are introducing a natural fertilizer to the plants as well, and this is allowing the plants to uptake the nitrogen and produce more biomass. And then the pots that didn't contain the microbes and biosolid pre-treatment, they had a much higher nitrogen level and a very low nitrogen level and a very high carbon level, which is not something that you don't want because the plants need to be able to have that nitrogen bioavailable to them and present in the soil. And then continuing with the results, I saw a significance in pots growing peas and broccoli together. And the pretreatment that was significant in this result was the presence of wheat, whether the pot had wheat or was not growing wheat. And so with the middle box plot, you can again see the same trend with this increased fluctuation and increased spread when wheat is present. And then looking at the soil respiration again, with pots growing wheat in blue versus pots not growing wheat in orange, you see that pots with wheat actually had a better establishment of the microorganisms. And at first this may be confusing, especially since you're thinking, wouldn't legumes have the major effect since they're in the symbiotic relationship with the nitrogen-fixing bacteria, allowing nitrogen to become available and supporting each other? But in this case, the possible reason for this huge difference when wheat is present is because of the root systems. Wheat is going to have larger roots than peas and broccoli and has more fibrous roots, so this allows more microorganisms to interact with the root system, which would increase the microbial population. So this is a good indication that it does matter what plants you choose. You're not only looking at, oh, this plant fixes nitrogen, but which plants have the larger root systems that are able to interact with more of the microorganisms and help with the soil. And so this is one of my favorite slides. So this is the regolith that we started with. And as you can see, as soon as you add water to it, it turns into cement. Such a wonderful thing. So I had to go through tilling and watering to go about doing that. But by the end of the experiment in December, it looked like this, which for the ecologist in me, it's like, oh my goodness. It's fascinating. It's just like, it really does matter what you add to the regolith stimulant to make it actually not only look like a soil you would find on Earth, but have the fertility and the characteristics that would be able to support plant life. And you can see that depending on what different pretreatments these pots have, and I forgot to mention there were 12 different pretreatments. If you want to learn more about it, we can talk after. That resulted in different colorations in the soil, and that indicates different nutrient availability, different organic matter, etc., etc. And so the conclusions for ecological succession, throughout this experiment, there were similar trends seen in the primary succession of ecosystems between my experiment and what has been done previously in research. And some of those papers I have listed here. If you would like to actually read those papers, I can send those papers to you or reference them for you. And so, major point, my experiment followed similar trends to what we see in ecological succession. And then in terms of a sterilized environment, since the regolith started off completely sterilized, there was an absence of competition with previous microorganisms that may have been in the soil before it was sterilized. And so by introducing new microorganisms and plants to the regolith, you are pretty much controlling how this ecosystem is going to go. And this can lead to faster establishment because of this lack of competition, which is one way to look at how we could expedite the process of ecological succession to form an ecosystem over time. And similar results in my experiment were also seen in this paper as well, done in 1979. And I couldn't find any other papers that were purposely looking at sterilized soil conditions, so this is actually a pretty new field. And then the implications of this study is that through looking at how we can form ecosystems from sterilized conditions, we can expedite biological feedbacks and cycle formations to expedite the process of ecological succession. We can better understand the formation of ecosystems, since it's important to understand that ecological succession is still a pretty, what's the right term for it? Controversial topic, because since we don't fully understand how ecosystems form over time, scientists, of course, are like, no, it goes this way, it goes this way. You need this, you need this. So this experiment gives us a better look at how these ecosystems could have formed over time. And then, of course, what I made at the beginning of the talk is increasing stability of quasi-closed ecological systems through following this ecological succession process. And then, of course, there's always uncertainties and possible errors that occurred in the experiment. So 30 of the pots were not comparable to the rest of the pots, the 90 pots in my experiment, because I ran out of time. And those pots were the ones that were growing the litter treatment. So in future experiments, I hope to actually have the extended time before I graduate and have to write a thesis to continue this experiment and actually test what a litter pretreatment would look like and how that may affect biological biomass production. And then not all the root systems were recovered, which affects total biomass collected. And it's because that regolith is so dense, you try to pull that plant out of there, it's automatically cutting off all the roots. So as you can see with this, this was a broccoli plant, and pretty much you only have the taproots there. And not all the taproots, just some of the taproots. So all that fibrous root material you can't really recover. And then, of course, this experiment wasn't done in a completely closed-off system, so it was exposed to unsterilized atmosphere. Of course, you have humans coming in, and they're all covered with microorganisms, so you have that as well. So there is a slight possibility that there was that influence as well. And so future studies to look forward to are looking at sterilized versus unsterilized ecological succession, further testing with different pretreatments to see how we go about forming these ecosystems, looking at multi-general microbe and plant studies to see if you can go through multiple generations of plants and form a soil from the Mars regolith simulant that is able to support indefinitely. Hyperbaric testing to test different atmospheric pressures and different atmospheric compositions. Regolith simulant characterization to see if we can find other simulants out there here on Earth that can become more publicly available for students and for other professionals who are interested in doing studies like this and make it more of a universal thing to research. And then species testing and selection, because you have to be very careful what species you plan to take to Mars. And so looking at not only what would be considered the typical Western plants like wheat, corn, potatoes, golden potatoes, but looking at quinoa, purple Peruvian potatoes out in the Andes maybe, things like that. And then looking at quasi-closed ecological analogs and building these analogs as well with deep space ecology. And then running human health and ecological risk assessments, which I'm looking forward to a lot. Just seeing how, if we build this system, how the human and ecological system may react to the technology you use, react to that interaction between humans and the environment, and things like that. And then deep space ecology has 3 test beds that we are looking at using. Let me catch up again. So for Earth, we are partnering with Emerald Kingdom Greenhouse to build our small-scale mock-up, small-scale prototype, and full-scale minimum viable product. And we are finalizing arrangements to build our small-scale mock-up at Oregon Eclipse 2017 this summer. Shortly thereafter, we are expecting some time in the fall to build our small-scale prototype, in which we can perform Earth-based experiments and technology tests. And then next year, Deep Space Ecology will come out with our minimum viable product. We currently have a team of 30 engineers and scientists working on system development, and we will soon be launching our technical advisory board of industrial experts. And then with the ISS and the LEO testbed, we are working with STARS, and some of you may have heard Heath Mills talk about his work with the ISS. He was on a panel yesterday, and we are working to bring some experiments up there. Then, of course, the moon, and we're in discussion with Ripple Aerospace to see if we can bring plants to the moon. That'd be interesting. And all of these test beds will help us be prepared when we go for Mars. And then just to finish up quickly, what does this mean for habitation in space? of course, the obvious thing is for exploration and for habitation in space, and to provide food for our Mars crew, but also to provide psychological well-being, to pretty much bring a piece of home with you when you're out there in such a barren landscape. And of course, providing a maximally adaptive environment for unplanned events. And then, of course, there's potentials for going out into space and building these systems, for helping here on Earth, how we can solve food security problems here on Earth by solving food securities in space and vice versa, and things like that. And so, acknowledgements, I had a lot of help at Duke University with funding, with advisement, with friends taking their time out of their day to fill 132 pots with Mars regolith simulant, and then Sire Industries for donating the Mars regolith simulant that I used in my experiments, and then additional thanks to Bill Abbey and Gregory Peters of Jet Propulsion Laboratory, who helped me get in contact with Sire Industries to obtain the regolith simulant, and then just having fun conversations and just fun collaboration on the project. Okay, thank you. Okay. Yep. Oh, what was the question again? Diagnosis. Yep. Yeah, and that's going to be coming from choosing what microorganisms you want to introduce to the environment that can help decompose plant material. And we're currently in the process of looking at different microorganisms beyond mycorrhizal fungi and rhizobium bacteria. Yeah, well, biosolids that you collect throughout your journey to Mars, that will definitely help. And then, let's see. I know an idea that I was talking with another scientist, was possibly starting plant growth while you're on your journey to Mars as well, so that you can have some organic material already that you could introduce into the environment. And also, before you even have humans going there, you can have rovers and other technology go up there to start building the